Hello everyone. My name is Young Choi. I'm a biology major student at UNC. This is my presentation for Bio 105L. And today I want to talk about what happens on a molecular level inside your cells during fasting, like starvation. So quick question. How many of you guys have tried the intermittent fasting or a 6 a.m. fasted cardio session because the social media told you it burns fat? We all know that it works, but have you ever wondered where that fat actually goes to in your body? It doesn't just evaporate, right? So to find an answer, we have to zoom in way past what the eye can see on a microscopic level. So today I'm going to show you the microscopic handshake that makes your morning fast actually worth it. So I have been looking at this research paper from Sarah Cohen lab here at UNC. So I'm not going to go through all of that, but basically they wanted to solve a logistics problem. Think of your body as a gigantic city like a New York City, Manhattan. Your fat is stored in warehouses, but it needs to be burned for energy in power plants, right? For a long time, scientists were not sure about how the fat got from the warehouse to the energy plant without getting lost. This study found the exact bridge that connects them, like a handshake. So let's meet our two main characters. The first one is mitochondria. Even for non-STEM major students, you might have heard that mitochondria is a powerhouse of a cell. It's because it's literally breaking down the fuel for our bodies to produce energy, which is called ATP. But you don't have to remember this for today's presentation's sake. And it uses fatty acids as a fuel source. You know, it's a main site of fat burning inside the cell. And, you know, when we run or walk for a long time, what we call a prolonged exercise, that's where the mitochondria uses the fatty acids as a main source of our energy, as like a fuel to produce ATP. So these mitochondrias are inside the cytoplasm of our cells, and it gets more active during the fasting or exercise. And here comes the second main character of today's topic. It's lipid droplets. It's basically like a bubbly, gigantic fat drops that floats around our cytoplasm inside our cells. So it stores extra energy in the form of fat, acts like a fat storage unit inside the cell. And you know, when it floats around, it gets attached to mitochondria to convey the fatty acid to from lipid droplets to mitochondria. So it keeps fatty acids safely packed when not needed initially, but then it releases fatty acids when it's needed for the mitochondria to produce ATP. So this is the actual microscopic picture of lipid droplets. You see the nucleus of a cell. You see little tiny gray, kind of bubbly circles. Those black gray circles here, these are mitochondria. And what we are trying to investigate primarily is that lipid droplets and the mitochondria forms a handshake and they transmit the fatty acids from the droplet to the mitochondria. So this is the essential sort of like a bottom line of what the paper is trying to say to us. Um, So the main molecule of interest is a plin 5. That's kind of like a hook structure. It's a protein that's attached to the lipid droplet side. And when they want to transmit the fatty acid, which is a fuel, from lipid droplets to mitochondria, the plin 5, the hook hand of the lipid droplets gets connected to the FAT4. That's the hook from the mitochondrial side. So they basically do a handshake. They form a binding, and from then on, the fatty acid is transmitted from the lipid droplet to the mitochondria. So think of like when we think about um like power plants, like how they works, the coal or the fuel is stored in the warehouse. And in our context, it's lipid droplets. And the power plants need this fuel to produce electricity, right? So mitochondria is the energy plant that's gonna get the fuel, which is fatty acid, from the lipid droplets in this context. Kind of easy, right? Um, Before we move on, I want to define a term that you'll hear a lot. It's beta oxidation. Kind of looks very complicated, but it's really simple. Think of your body as a campfire. You have these massive logs of wood, like the fuels, the basically the fatty acid. Those are your fat stores. But you can't just throw a whole 50-pound log in a teeny tiny fireplace and expect it to burn just, normally, right? You have to chop it down. You have to chop the whole log of, woods into smaller pieces, right? So beta oxidation is basically the cellular X, the cellular process that breaks down these gigantic woods into smaller pieces that we can actually use it for to throw it into the fireplace. This is a process of chopping those big fat molecules into tiny pieces so the engine, in this context, mitochondria, can actually burn them for energy. So when I say beta oxidation is increased, just think that the cell is successfully chopping and burning its fat fuel. But in science, observing a connection is only the first step. To prove this handshake, like I showed you before, um, the research team, the Sara lab researchers, Cohen lab researchers, had to provide physical evidence. So now we're going to look at the four core assays experiments the Cohen lab used to prove that these proteins are not just sitting there, but are actively moving fat to be burned for energy. So the first assay that they did is called fluorescence microscopy. Again, it sounds very complicated, but let me break it down for you. Imagine putting a neon glow in the dark sticker on a warehouse, the lipid droplets, and a bright red sticker on the engine. So that's the mitochondria. In normal cells, the green and red spots were all over the place. But in the starved cells, the hungry cells, the spots overlap to create a yellow glow. Um, so the key takeaway is if you take a look at the figure D, um, so here's the control. And so basically the plin 5 C delta is just truncated plin 5 molecule without tether domain, without the actual hooking site. So if you take a look at the normal plin 5, um, the pink area is the mitochondria and the green area is the lipid droplet. They are vigorously overlapping to each other. But if you take a look at the truncated version, the chopped off version, which does not have the actual hooking site, connection site, as you can see, it's not overlapping pretty well compared to the normal plin 5. So that's the key takeaway here. And figure E is just a quantified data for figure D. This proved that fasting is tr the trigger that forces the fuel and the engine to physically touch. So as you can see, these the numbers here are just different types of truncated tether domain of plane 5, and this plane 5 is just a normal plane 5 with the actual normal functioning connection side. So this what this is trying to tell you is that the truncated tether domain, if the plane 5 does not have it, the overlapping, the handshake doesn't really happen. So for a normal handshake to happen on a molecular level, they need a fully functioning plane 5 molecule with the tether domain on it. That's why the co-localization here, the normal plane 5 has the highest value, almost like 100. So basically more co-localization means more handshaking. That's the first experiment that they did here. Second, they wanted to know um, if the the protein that the plin 5 is interacting with is specifically FAT4. So what they did on a figure A here is that they found all the potential candidates for mitochondrial protein that could actually connect with the plin 5 protein on the lipid droplet side. So from, ZNF 598 to GIT1. These are all mitochondrial protein candidates that could be used when combusting fat on the mitochondria. Insane score is that the higher the value is on the sane score, that means there's a higher probability of that said molecule, that said mitochondrial protein being the partner of the plin 5 molecule. So as you can see, here I see the FAT4 being one of the candidates, and it's got a zero point 0.9 sane score, which is pretty high. Um, and these are what the figure B here is saying is that um so the in the previous slides, these are the truncated versions with truncated tether domain of plin 5. So the lower the number, the larger the truncated portion of the protein. So what the number here is trying to tell you is that the longer the tether domain is truncated, the lower the protein folding equals lower colocalization, lower transfer of um, fatty acids from lipid droplets to mitochondria. So figure C is the called Western blot is one of the most popular experiment that a lot of biologists are exposed to, even the UNC undergrad students like me. You're very familiar with this technique, and it's just about finding a specific protein, locating a very specific protein inside the cell. Um, So think of our our cell has tons of millions of proteins inside the cell. And for us to locate a specific cell, like for example, for in this context, plin 5, we are using Western blot. So what you have to pay extra attention on here is the elution part, and you see the um blurry three lines over here, and on the left column it says F84. If there's a band appearing on the elution side, that means we successfully located the FAT4 protein on the Western blot. So think of this as like a face ID for detecting proteins. To find our specific hook protein out of the thousands of millions of protein inside the cell, we use a special glowing tag that only sticks to that specific plin 5 protein or FAT4 protein in this case. And when you see a thick black line on the screen, like here, that's the Face ID confirming, yes, the hook is there, and here's exactly how much of it we found. Excuse me. So this is our way of proving that the proteins we are talking about are actually present at the scene. So the third experiment they went on to they did is called pulse chase assay. So you can think of this as like putting a GPS tracker on a piece of fatty acid to see where they are coming from and towards where they are moving to. So from starting from, lipid droplets like we discussed before to the plin 5, the hook of the on the lipid droplet side to the FATP4 which we just detected from the Western blot, the hook side of the mitochondria, and then ultimately to the mitochondria. So we are tracking all these movements of lipid drop fatty acids from start to end. That's the gist of it. So first, the Red Sea 12 here is just another name for fatty acid. CM is just normal condition. 4 hour HBBS as is basically means that the cell is starved for 4 hours. So we are comparing these two different conditions. And this is where things get interesting. Under normal conditions, the CM, the labeled fat mostly stays in the lipid droplets. In other words, the fat just sits there in the storage on the lipid droplet side. But once the cells are placed under starvation conditions, the 4 hour HPSS here, we start to see the shift of fatty acids to the mitochondria. The fluorescent signal moves away from the lipid droplets to here, and gets closer to the mitochondria. That means that the fat is no longer just sitting there and has moved to the mitochondrial side. The way the diagram here looks is kind of counterintuitive because at first I thought there's more fatty acids residing in the lipid droplet side for the starvation condition, but it's actually the opposite if you take closer look. So this movement of fatty acid doesn't happen in any situation. It happens specifically when a cell goes through a starvation, and that's where this whole complicated story connects to our real life example of intermittent fasting and so on and so forth. Yeah. And then we move on to our last experiment, which is radioactive assay. This is where the beta oxidation part comes into play. So finally, they had to make sure that the engine, the mitochondria, was actually running. And one way to test it out is the beta oxidation, which is cutting down the fatty acids to efficiently combusted to produce the ATP. And the And the axis title here, it says efficiency of FA flux. That just means the efficiency of fatty acid flux, how well the fatty acid is flowing and burning inside the mitochondria. And I just wanted to make a point that here is, it sounds like a gibberish, but it's really simple. It says it's S155E. That's just the activated version of plin 5. So in this case, just the normal plane 5 without any, S155E added, that is just inactivated version of plane 5. This is activated version. And this might sound familiar to you, right? 5C delta, that's the truncated plane 5. Um, yeah, so they measured the exhaust coming of the mitochondria when the handshake was strong. Um, the exhaust was high. So the strongest fatty acid flux efficiency comes from non-truncated and fully activated version of plane 5, which is shown in the blue triangle here. Other conditions have shown almost zero uh, relative FA flux efficiency. So what's the big takeaway for your next workout? Fat burning is not just a vague concept, it's a physical delivery system happening on a molecular microscopic level between mitochondria and lipid droplets, as we discussed. So first, fat burning is a delivery system. Burning fat is a physical logistics process. For fat to be used for energy, it must be physically moved from storage, the lipid droplets, to, yes, the pLin5 to FAT4 to mitochondria. And pLin5, the hook of the lipid droplet, is essential bridge for this. Without this connection, the fuel cannot reach the engine. Also, like we've seen the previous slide, the activation of pLin5 is important for fat combustion. Only when the switch is on does the delivery of fat and the energy production speed up. And lastly, the science of your workout. This study provides the molecular why behind fitness trends like intermittent fasting. It shows that we are quite literally flipping a cellular switch to allow our bodies to access and burn stored energy more efficiently. Thank you so much for listening.